very strong group of people, development of novel agents in the setting of sarcoma, along with several other diseases. With that, again, a few things as regards disclosure. And so I'm doing this as part of a company. That company is called ZIOPHARM Oncology, and I lead that company. I work with the Sarcoma Foundation of America. I'm funded by the Kristen and Carr Foundation. And formerly, many years back, I worked very closely with Genentech, again involved in Roche, in several drugs that have evolved. For example, IGF, along the pathway with sarcoma. Now, in contrast to what you just heard as regards the role of the surgeon, so I think in metastatic disease, there does come a time when surgery has a much more limited role, although I'm still actually not quite sure about that. And the reason I'm less sure about that is that our understanding of cancer is evolving tremendously. And what's evolving is that clearly there's something wrong with the primary cell. We know that. But it's not just that cell. It's a system that's gone awry. And so, in fact, what's going on is that the primary gives rise to metastases, but certainly in other cancers. So breast cancer, I think, is one that we understand as regards this mechanism, but the metastases are giving rise to other metastases, and actually after the metastases can feed back on the primary. So the role of the surgeon, I think, is still there, and I think will continue for some time. And so I'm often reminded of a story I heard about Dr. Brennan. This was at a time when Dr. Brennan had first come to Memorial Sloan Kettering. The physician-in-chief at that time was Dr. Vita. I'm sure a lot of you are familiar with the very large textbook in cancer, Vita, Rosenberg, etc. And so Dr. Vita had been a big proponent of the idea of extreme high-dose chemotherapy. And at that time, looking forward, kind of analogous to us now, looking forward, for example, say with targeted therapy, Dr. Vita and pretty much everyone believed that this was going to be the cure-all for all cancer. It was very logical. It made sense. What they were doing was transplanting everyone, breast cancer, everyone, with extreme high-dose chemotherapy. And so he posed the question to Dr. Brennan, and I may have it a little bit wrong. I mean, I don't have the exact thing. But anyway, what would Dr. Brennan do somewhere around about 2000, the term millennium? Of course, there was a good definition with time. What would Dr. Brennan do when there'd no longer be a role for surgeons because the chemotherapy, the high-dose chemotherapy, was going to be curing everyone? Of course, 2000 came along. Dr. Brennan was still operating, running 2 days a week, running 2 rooms at the same time. I don't know how many cases he was doing, a lot. And Dr. Vita was no longer at Sloan Kettering. And so I would argue that in this disease, and I'm obviously looking at primary disease, but I'm not certain about this whole thing, that probably the most effective small molecule to this day still is the atomic element iron. Okay, that's a small molecule, and it's iron in the setting of the steel of the surgeon's knife. And again, as you just heard, very elegantly articulated, it doesn't really matter whether you use a knife or any other kind of instrument, but it's that resection, okay, that's very effective in treating these solid tumors, many solid tumors. All these other things that we're doing are effective, and some of them are very effective. I'm still struggling as to how effective they are. And I'm very fortunate to work with a team of people who are very, very good. And that team includes in sarcoma some of the best experts in the world, in breast cancer, people like Larry Norton, in testicular cancer-related people like Larry Einhorn. I mean, true experts in the field. And so I personally struggle with what is the best pathway forward, except to say that I think there's several pathways forward. I'm becoming, that's why I like the slides that Dr. Brennan put up this morning, I'm becoming less biased in one direction and more that I think we need to be open to several directions. And so what I'm going to articulate now are really my biases, which hopefully are more open in terms of several directions. And again, I'm going to try and avoid getting into too much specific detail, except for a few things, again, biased towards me. So research and development, there's really 2 areas. So research, and this is driven by increasingly in the world of developing new drugs, biology and engineering. And biology, of course, is, as I said, the cancer cell, the environment, and engineering is having a much larger role now. In fact, it's huge. There's a revolution that's going on right now. It started within the last few years, and it's just accelerating tremendous. It's exponential, like nothing we have ever seen before. In fact, it may be a time right now analogous to when Ford hit the automobile industry, because automobiles were being made in a bespoke fashion. Ford came along and he started doing things where all of a sudden the price came way down, and more were being pushed out. And I think the same or similar may start happening in terms of how we do drugs. It's going to take a few years, but I think we're starting that right now. The biology is key. We heard that before with surgery. It's key with all of this, and it's key with the cancer cell itself, so the things we're targeting, but it's not just that. It's the environment as well, and we cannot forget that, because as I just mentioned, it's very clear, for example, in breast cancer, it's very clear that what's going on is there's a dynamic between the primary tumor and metastases, and it said there's 5 sites of metastases, that cells are moving between the primary and the first site, the primary and the second site, between the first site and the fifth site, and the bone marrow is involved as well. Okay, that's site number 6, and it's not really a metastasis, but it's a safe haven for cells. And it's critical to understand. So we can target one target in that cancer cell, but the likelihood of success is very, very low unless it's a crucial target in the biology of that cancer. So Gleevec is a remarkable example in GIST. GIST has been, again, it's a hallmark of this, like CML. But there's not too many other examples like this. There are some actually in sarcoma, but again, these are rare subsets of a rare disease. And we must remember that as we move ahead, just in terms of trying to be practical with this. So the development part, and that's really the clinical trials. It's a few things. It's the clinical science. And again, that's not easy, but everyone's trying now to make this better and better. It's not easy because the science part in of itself is difficult. And then, of course, we must deal with the regulatory authorities, so the FDA, the European authorities, and elsewhere. And sometimes the goalposts shift, and they're doing that right now as an example with sarcoma. Sarcoma was thought to be it is a rare disease. At least I think it's a rare disease. We all do. But it shifted recently at an ODAC meeting. ODAC is the Oncology Drugs Advisory Committee, and at that meeting, the oncology leadership said that sarcoma is a rare disease, but it's not an ultra-rare disease. And so it became a little bit harder now, more difficult to get drugs approved in sarcoma. So it keeps on shifting. And then again, there's this thing here, which is the bottom line. Okay, and the bottom line is that in a world where economies are bouncing back, and they are bouncing back, but at the same time, we need to be very clear here that the amount of money coming to the pot is less and less. And so this is reality, that there's a cost that goes along with this. There's a commercial opportunity, and this is looked at critically by big pharma, by investors, by everyone involved in these equations, and there's got to be a justification. So I have to be able to stand up in front of the people who are paying for all of this, say, this is what I think it's going to cost, this is how long I think it's going to take, and I'm usually always wrong because it's always going to take longer, cost more. So, and they know that, but the reality here has got to be built into this, and what is the commercial opportunity? And so with a disease like sarcoma, it's not always that easy because it's a rare disease. But there may be opportunity here. And so it's a rare disease. So, and we know the incidence in the United States in terms of soft tissue in adults is somewhere around about 10,000. And I think it's important when one's developing drugs that we need to break it up into certain subsets and then make it as homogeneous as possible. And I think, for example, we have certain drugs that work both in soft tissue and osteo, but for the purposes of running a very good randomized study, it's often better to leave out osteosarcoma in that setting, bring it in later on, when a randomized setting, and leave out certain subsets so that you get very biologically homogeneous groups. So there can be no arguing when one goes to the FDA or others about any kind of bias between the two groups. So one often leaves out certain subsets, get to a very biologically homogeneous group. But understand, again, this is rare. It's very rare. It's heterogeneous, and one has to, anyone designing these trials, you've got to offset that. And often the offset is to leave out some of the rarer types that we know could cause wobble unless they're perfectly balanced between both groups. And in running a randomized study, it doesn't matter how big it is, so it can be 400, 500, 600 patients, one still has to keep somewhere around about 2 strata for that. So there's no way you can start including osteosarcoma, various other rare subtypes and so on. So the most practical way is just to leave them out and then include them later on. So keep it as homogeneous as possible. So clearly there's inefficiencies here. Obviously there's a lot of challenges with this, but at the same time, if one does this in a very rigorous way and applies certain science to this, it could well be an opportunity. That opportunity is several-fold. And so, as I said, the goalpost has shifted with the FDA. That happened somewhere around about 3-4 weeks ago at an ODAC meeting. And so, from the FDA perspective, the FDA definition now for rare disease or ultra-rare diseases, we speak about cancer now, is that it needs to be less than 1,000 per year. So sarcoma, which did fit in before, as a whole group, does not fit in there anymore. If we go after rare subtypes, it will still fit in. But there are ways of wiggling through that, and we as an organization have certain people who work with us who are very facile with this, and that is absolutely key and fundamental in terms of developing this and developing a plan with FDA, with the European authorities as well. And again, this has got to be measured here against what this is going to cost, and again, the investment return that's going to come back to the people who are going to pay for this. Because as compared to a lot of lab studies, which are expensive by themselves, the cost of clinical trials are often an order of magnitude or orders of magnitude more. And so it depends exactly what, how, and so on and so forth. But to me, the average cost per patient of running a pivotal trial is somewhere of the order of $100,000 per patient. And so if one calculates that out, a 400-patient trial is going to cost somewhere around about $40 million. So one has to be very sure going into that, one, that the likelihood of success is high, two, that whoever's going to be paying for that, and it's often me standing in front of those people, that I can justify to them the rationale as to how and why what I'm doing, and this over here, that there's going to be a return in terms of how this is all going to work. So let me take a step back now and go back to a slide that was shown this morning by Bob Maki. And the way that Bob has articulated the galaxy of sarcoma is really into 2 at this time. And it's the world of GIST and really other. And I think that's a reasonable way of doing it. Actually, I think it's pretty good. Because I do think GIST are quite different, and I think the others, even within the subsets where we know what a lot of these targets are, I think they're very different. And I think it's really important, I'll get into some of this in a while, because it's again the idea here of necessary and sufficient. But GIST is an example like CML, where going after one specific target here has been absolutely pivotal in terms of that impact. The likelihood of that happening in other cancers is probably low. We don't know yet for sure, but it's probably low. And it gets onto this idea here of how many different targets do we actually have to hit to have an impact on a cancer. And so one target is often necessary, but it's probably not sufficient. We're talking about the non-GIST galaxy now. It's probably not sufficient. How many are sufficient? I don't know the answer to that, but probably many more. And that's where the idea of combination therapy comes along. And again, the basis for this is that a lot of these are systems that go awry. So it's not just one gene in most cancers, but it's a series of gene networks and gene systems. And so developing drugs there becomes much, much more complicated. A lot of the big pharma companies are starting to do this now. And they're doing it with targeted therapy. And so, again, not just using one, but using 2, 3, 4, and 5 at the same time. But it's much more complicated. and difficult to apply to a field like sarcoma. So clearly more complex, okay, probably much, much more complex. This is what we do, and this is where sarcoma fits in. So the first here, I'll get into some details shortly, is we're doing a randomized phase 3 study, a pivotal study, with a molecule called Palifosfamide. The second here is in an area of synthetic biology and several different aspects here, applying this, and probably going to apply this to some of the rare subtypes of sarcoma. So with that, let me get into some detail, again, showing you this in a portfolio fashion and focusing down on Palifosfamide. So on the one hand, doing something that is extreme in terms of being at the absolute cutting edge. So synthetic biology, and I'll get into a lot of detail shortly, but it's using the absolute engineering power of synthesizing DNA and then using DNA as a therapeutic. And the power of that as a therapeutic and the potential of that seems to be immense. Conversely, and let me tell you the story about this briefly. So Bob Gale, and Bob Gale is well known in the area of leukemia and bone marrow transplantation, used to work with us. Way back, he was working with a group at Southern Research Institute. That is a part government group, part government, part UAB, University of Birmingham, Alabama. And he was working with them looking at different drugs in terms of bone marrow suppression as regards bone marrow transplantation. And as he was doing that, they were looking at cyclophosphamide, ifosfamide, and the one group there who've been very good came up with a drug, and what they'd been doing was looking at the drug ifosfamide. In any event, this, as I was looking at this, linked up with sarcoma because of ifosfamide. And what was interesting as well was that this group was very good. They're a preclinical and a discovery group, but firstly out of that building at SRI, Southern Research Institute, I think the number 7, it's either 6 or 7 different drugs have now been FDA approved in cancer, and I think that's as much or more than any of these large pharma. So they're very good large pharma companies, and then this one specific group have been very successful in terms of what they've been doing. So they really know what they're doing. And we went after this again with the idea here that ifosfamide is a good drug in sarcoma, but just a very difficult drug, very difficult. In applying new understanding of biology and new molecular components with this, we could potentially make this a much better drug in many aspects. And so what this is, palifosfamide, really is the active warhead, if you will, of ifosfamide. So it's the active metabolite. It's very similar in many respects to bendamustine, very similar to cyclophosphamide. And the whole reason to do this, and we've done a lot of preclinical work, I'll show you some of it, but it in many respects is more effective. Clearly the toxicity is much lower. In patients, the quality of life is a whole lot better as compared to ifosfamide. Much easier to administer. It's an outpatient drug as compared to inpatient with ifosfamide. And it's cost effective, and we've not measured that exactly, but the amount of time in hospital is much less, and the complications are much fewer. The first way we want to do this is in soft tissue sarcoma, and again, I've got to justify this to the people putting the money in, and that's a niche indication, but we think this is going to work in other cancers, and we're doing that now at the same time in small cell lung cancer, and we're doing it with, again, this person, Larry Einhorn. Einhorn's very good because he worked early on with that one, ifosfamide. He actually got that FDA approved in testicular cancer. So he's the Lance Armstrong Professor of Oncology because Lance Armstrong is his patient because of a lot of the work he did in testicular cancer. So he's helping us a lot with this. He's got a lot of understanding here. We've got an oral form here as well. And again, it's Southern Research Institute together with us developing. And what this is, again, it's a small molecule, and it's a small molecule that effectively cross-links DNA. And it does it in a way that is just much less toxic and we think more effective as compared to ifosfamide. So it's not a prodrug. There's no need for any enzymatic or genetic activation. As compared to ifosfamide, the hemorrhagic cystitis and CNS toxicity, there's zero because there's a clear molecular reason. Those metabolites are not present with this drug. There's no need for MASNA. In preclinical testing, we're doing this with Rich Gorlick and many others. We've been doing it in the lab setting just to test for preclinical farm tox and with a view to going into pediatric studies. But again, in preclinical studies, it's active in Ifos and cyclophosphamide-resistant xenografts, including many sarcoma subtypes. And again, we're looking to have this not just IV, but oral as well. And there's many reasons why we like oral. In sarcoma, in the pediatric setting, and then we're looking at other indications as well, not germane to right now, but in breast cancer, we think it would have a lot of applications as well. So a lot of preclinical studies done here in order to move this along. They're listed over here. And as we did this, the idea here was, how do we get this into a sarcoma treatment? And the bottom line here was that this should be combinable with adriamycin, so with doxorubicin. Again, broad activity seen just in many cell lines, xenografts and so on. This was an experiment from Gorlick, Gorlick, who works both in his lab and in several other labs. And this is being done as a feeder into ultimately the Children's Oncology Group. But the bottom line here is that in resistant cell lines and resistant xenografts to cyclophosphamide and to ifosfamide, there's clearly activity. And that's obviously a very good thing, especially in the pediatric setting, because in pediatric sarcomas, these drugs are used extensively and a lot of resistance happens. In adult sarcoma as well, we think it's going to be, again, very helpful. Same thing seen here, again, in the setting of osteosarcoma. Again, this drug, as I said, is orally active. We're working with this extensively in the setting of breast cancer, but we want to do this in sarcoma. Same thing over here. We did this with Larry Norton. And Larry Norton is a big proponent. He's a breast cancer doctor, a big proponent of oral therapy in breast cancer for many reasons, one of which is that he does mathematical modeling where dose and schedule are very important, especially the schedule. So being able to give a drug on for a certain amount of days, off for a certain amount, and the effect seems to be a whole lot better with this on-off rhythm. And that's exactly what we're doing here. And we want to do the same thing in sarcoma. And again, the same thing shown over here. This is combining us here, again, with doxorubicin, with adriamycin. We did this in the clinic. This is done by several different centers. This was presented by Monica Mita from San Antonio. And this is a patient with rhabdomyosarcoma. And just to point out very graphically, before, during, and after. And so a lot of different signs of drug activity. And the net of this was that we did a randomized phase 2 study. And one of the things I've learned along the way is that, in general, when one can, a randomized phase 2 study is very good to do before going into a full-blown randomized phase 3. So in other words, put it the other way around, non-randomized phase 2 are often very difficult to interpret, whereas randomized, a well-run randomized phase 2 makes it much easier to get a sense of predicting what the randomized phase 3 will do. And so this was done in a very rigorous way, stratified by age and certain histologic subtypes, with assumptions as to other components here that would balance out, and they did. So this was a very well-balanced study. This was presented at ASCO this last year by Claire Verschragen. This involved, I believe, about 15 different sites, of which 10 were actively enrolling. Claire is the head of oncology at the University of New Mexico, soon to come up here to Vermont. And the bottom line from this, looking at PFS, was a hazard ratio of 0.43, which was significant, and a difference in terms of the median PFS of 3.4 months. And obviously that was meaningful, it was clinically meaningful, and is the basis for a randomized phase 3 study. And to that end, just in terms of looking at this, to our knowledge, we've looked at many things now. There's no other drug that's done this before in sarcoma. Now, there's many caveats. So this study was not designed to look at survival. So although we know that survival was trending in the right direction, we really don't have information on survival, and we won't have information on survival. And to FDA, survival now has become a key endpoint, although to a lot of sarcoma clinicians, especially in front line, this is very, very meaningful. If we can do this again, most people believe that this drug will get approved. We showed this as well, and this is a Kaplan-Meier curve, just again looking at patients getting less than or equal to 6 cycles, and then censoring, and again the take-home message here, a hazard ratio of 0.39, about 0.4. And we've used that as a basis to go into a pivotal phase 3 trial. And again, people like calling it these fancy names. It's a good name because I remember the name PICASSO. I honestly can't remember what it stands for, but I do remember the name PICASSO. So it's a 424-patient study running right now, comparing it to the standard of care, which is used at most places in the United States, Canada, Europe, and the rest of the world, and that's frontline Doxorubicin here with the placebo versus Dox plus Palifosfamide, set up for PFS for accelerated approval, OS for full approval, and looking for a hazard ratio of 0.60. So we showed 0.40, and we're looking to show 0.60. And again, we think if we are able to do this in a very rigorous way, and that's an if, although we spend a lot of time making sure it's being done in that way, it's probable that this drug will get FDA approved. And so this is well underway right now, and we'll get to data the middle of 2012, the middle of next year. With this, just to briefly point out, we're working with Larry Einhorn. Einhorn knows ifosfamide extremely well. He got that FDA approved in testicular cancer. We're expanding it to small cell lung. He's treating patients with testicular. It's early days yet, but he's seeing the same with similar things going on in both of those settings, testicular and small cell lung cancer. Let me use the last 10 minutes, last few minutes here, just in terms of synthetic biology, and tell you that this is an area here of great promise. We recently combined with another group to do this, and you can read, this is from The Lancet, a very conservative journal. This is an editorial from the beginning of this year. And I think the reason that they think that this is going to change so many things is, as I see it, back to the Henry Ford analogy. And so all of a sudden, as compared to a random way of doing things, it's an engineering process that's being applied. And so I think the vision, certainly that we have, I have, and others have here, is being able to lower the cost, because that's fundamental, is lower the cost, be able to do things much more quickly. So instead of having a drug that comes on and then runs for 20 years, perhaps after 5 years, get the new version of that drug. There's no reason we shouldn't be able to do that. And again, with this, be able to get things through in a much quicker way. So the discovery part quicker, hopefully we can mirror that with the clinical part as well. And so, again, I don't want to go into too much detail here, but many components here that are key, having a part component here where off the shelf one can get all the different components, be able to rapidly synthesize that. Again, the bottleneck to this is the current regulatory system, which, as it should be, is very much focused on safety. Again, one's got to get to the IND, the investigational new drug component, and the next components. So it's going to take time with that, but I think as the system gets going, we'll go faster and faster. And I think as we look at this, for the setting, for example, of these rare sarcomas, because practically it's very difficult, I think, for any large company to develop new drugs in very rare sarcomas, unless one applies a system like this, where one has very rapid throughput with an engineering type system, and one can get to an answer, yes, no answer, very quickly, so you don't spend huge amounts of money and then get that return going. This is a very good way to approach rare disease, not just sarcoma, but other very rare cancers. That is our underlying fundamental approach to this. I don't want to go through all of this with you, except to say that with this as well, we have different ways here of being able to lock onto proteins inside cells. And so one of the difficulties, for example, we heard about KRAS earlier. KRAS is a very good target in oncology, but it's been very difficult to drug that target because it's a protein-protein interaction. So no small molecule to date has really worked there. This may be a way to avoid all of that and use a protein-protein interaction. So we're busy working on that. We're working on several different things with this. And again, ideally, if we can get this right, I don't know yet, we've just started, if we can get this right, we can go after these very rare cancers, sarcoma and other ones as well, because we can get the cost way down here. We can justify to people going after this because the cost going in is very low. So again, this idea of protein-protein interaction, and the idea here of, instead of building antibodies in a large vat and spending billions and billions in terms of building that manufacturing facility, instead just bypass that use our own bodies to make those antibodies. And there are many ways of doing that. It's no longer science fiction. This is something that I think will happen in the next few years, and again, applicable especially to rare cancers, like subsets of sarcoma. We can go after certain targets and produce antibodies and or these protein decoys that will be relevant, get inside cells at a very, very low cost. Let me conclude and come back here again. I think the era here of targeted therapy is a reflection that we have come such a long way in terms of understanding biology. That is fundamental to being able to move forward. I think the practical connection onto that is almost there, but maybe not quite there. It may be, but I don't know. But it may be analogous to the conversation that Dr. Brennan had with Dr. Vita, and I wasn't there. I've heard about this third or fourth hand. But it may be analogous to that conversation sometime during the 1980s about the death of surgery and chemotherapy that was going to cure everything at the turn of the millennium. So I think that targeted therapy used in a rational way is going to work. I don't think it's going to cure everything. I think it's a combination of many things here as we develop different programs, different ways of doing it, that likely are going to keep on moving the needle, because that needle is moving. It's done so in other cancers, testicular cancer, leukemia in children. It's done so, I mean, if one looks at GIST tumors, that's a huge example. It's going to happen in these other ones as well, but it's going to happen when we combine all these different things together. I want to give you just one quick example here, because IGF is something that I got to know a lot about, these IGF receptor inhibitors, which by all accounts should work gangbusters, but of course none of them, not one has worked. But there is a drug here that does work. Okay, this drug, I don't know how old this is, but it's old. It predates, way predates Dr. Brennan's conversation with Dr. Vita. So I think it goes back to the 1950s or 60s. I don't even know. It's old. It's used as an oral hypoglycemic. There is, I don't know the exact amount, but it's somewhere, I believe, north of 50 million in terms of NCI funding going into this right now, because it has such a profound anti-cancer effect. And the cost of each tablet is pennies, or less than pennies. I think it's pennies for like 100 tablets. And the anti-cancer effect is obviously a direct effect in terms of blood sugar, but then a lot of these indirect effects here in terms of insulin growth factor, mTOR, we know about that in terms of other sarcoma drugs, NF-kappa B, cancer stem cells, and so on. And I think, again, it's just a reflection, at least to me, philosophically, of being open-minded. Clearly, there's a lot of biology we're learning so much about now, and there's going to be more and more and more with high-put screening, computational biology, systems biology, and we need to be as open-minded as possible, because we already have so many things that I think are going to help us enormously. Here's one that is very old, very inexpensive, and I think is going to have a huge impact. So again, let me conclude. These are rare tumors. Everything you look at here is really wrong in terms of drug development. As we look at it, obviously I am biased because of my experiences, but I have been able to justify to some of the most sophisticated people, investors who give out money, and they're very careful how they give it out. I have been able to justify to them over and over and over the opportunity that exists here. I think everyone sees that, and it's a unique time right now. I don't know exactly yet how it's all going to unfold, but I am pretty confident it is going to unfold, and it's going to happen soon. So the next several years, the next 10 years, as we take all the biology we're learning about now, and we're going to learn more, and translate that into new treatment modalities and new treatments. Again, thank you very much, and thank you all very much. Thank you.